Hello, I'm Dr. Francine Foss, and we're here at iwNHL 2015 at Lake Maggiore. And I'd like to introduce the members of our discussion on T-cell lymphoma. Dr. Del Mori, to my left, is also one of the co-chairs of the session. Dr. de Leval spoke on molecular mechanisms in T-cell lymphoma, and Dr. O'Connor talked about interesting new combination therapies. Let's start off the discussion by talking to Dr. de Leval, who gave us a very elegant presentation about some of the new molecular genetics and new genes and mutations in aggressive T-cell lymphoma. Can you talk a little bit about the most significant findings and how that's going to lead us perhaps to a different therapeutic approach for these diseases?
Yes, so there has been a lot of recent discoveries in the field. Until recently, very few recurrent translocations were characterized in peripheral T-cell lymphomas, essentially in ALK-positive anaplastic large cell lymphoma. There is no evidence that there are recurrent aberrations not only in ALK-negative anaplastic large cell lymphoma, but also in other peripheral T-cell lymphoma entities and T-cell leukemias. So the interest of those is that they target some molecular pathways and are potentially amenable to inhibition by small drugs. More on the biological, from a biological standpoint, the interest is that these molecular aberrations appear to correlate with some aspects of the T-cell lymphoma entities, such as the cell derivation. They also help to refine the diagnosis, as some translocations, for example, in anaplastic large cell lymphoma ALK-negative, are associated with distinct clinical outcome and may help stratify patients in future clinical trials. As for the molecules that are often mutated are various elements of the JAK-STAT pathway. STAT3 and STAT5b in particular are often altered, and there appears to be some of a correlation between STAT5b mutations and either homogeneously aggressive PTCL entities such as prolymphocytic leukemia or hepatosplenic T-cell lymphomas, and while the STAT3b aberrations appear to be more common in more indolent T-cell lymphoproliferation. And finally, among the pathways that are recurrently targeted, the various intracellular ways downstream the TCR signaling appear to be targets of alterations in nodal T-cell lymphomas, including the angioimmunoblastic T-cell lymphomas and related entities.
You also talked a little bit about some of the earlier mutations, the epigenetic modulators and some mutations that may occur early on, particularly in perhaps even in stem cells. Can you talk a little bit about those and how they perhaps could be impacted by some novel therapies? I think that will impact on the discussion that we'll have when we talk with Owen.
Yeah, sure. So the epigenetic gene modifiers are recurrently mutated in angioimmunoblastic T-cell lymphoma and related entities. These genes are TET2, DNMT3, IDH2. Although the biology is not fully understood, those mutations probably alter gene methylation and expression. They suggest that these tumors may be amenable to therapy by demethylating agents. As for the biology, again, the interesting finding is that some of these aberrations, in particular TET2 and DNMT3 mutation, may be present not only in the T-cell lymphoma, but also in precursor cells and precursor above the myeloid to lymphoid separation, so really hematopoietic stem cell lineage, suggesting a multi-step way of T-cell lymphomagenesis. Additional secondary alterations, maybe, for example, ROA mutations that are rather frequent in that entity.
So that was an exciting finding, the identification of ROA, and you mentioned that that's a dominant negative mutation.
Yeah.
It's also found in other types of T-cell lymphoma?
So ROA appears so far to be rather specific for angioimmunoblastic and TFH-related lymphomas. It is not specific for T-cell lymphomas. Other ROA mutations are also found in solid tumors like diffuse type gastric cancer. Some of them have also been described in Burkitt lymphoma. I must say I have not worked myself on ROA biology, but from what's available in the literature, it appears to be clear that the mutant protein has a dominant negative effect, but how to connect this to lymphomagenesis is currently not clearly understood.